welcome to 15.1, our lesson on fertilization. We are looking at these very important terms. Gametes are sperm cells and egg cells, each with 23 chromosomes. We know that that will allow for that first cell of life to have 46 chromosomes, and that's the cell that will divide many, many times during fetal development. So that's what you started from, is a 46 chromosome zygote. And the embryo is a term used for about 2 months, first 8 weeks of fetal development. And in that time, all the body structures are forming. And when that is completed, around 8 weeks later, and then for the rest of the time of the pregnancy, it will be called a fetus, and all the body organs are intact and just growing bigger. Fertilization is also called conception, and that is what happens generally right after ovulation up to 24 hours. And the egg is viable at that time. It will happen in the fallopian tube almost all the time, because that's the spot where the egg is when it is viable. And after that, the egg doesn't survive much longer, so the sperm would have to meet it at that time. Now, there are many hundreds of millions of sperm that can enter and head towards the egg. However, because half of them are going up a fallopian tube that there is no egg in, that leaves only half of them. A lot of them are going to perish because of the acidic conditions in the female, maybe even acidic conditions in the urethra of the male. And so there seems to be only a few hundred that survive when it is the time that it reaches the egg. Here we see that the egg lives around 24 hours, maybe up to 48 hours, so a day or two is the only window of time that the sperm has to fertilize that egg. You will not have to know all the names of the coatings around the egg, but the zona pellucida is one of 3 layers that surround the egg as protection, and it is through this layer that the corona radiata and the zona pellucida it is through these layers that the acrosome of the sperm containing enzymes is needed, because it will take probably 500 sperm worth, it looks like only one in this diagram, but it takes probably 500 sperm worth of acrosome enzymes in order to make this tunnel through the outer coating of the egg, and actually allow, at some point there'll be one tunnel that is able to get a sperm in first, and that first sperm that can get into the egg will be the one that releases its nucleus, and that nucleus will fuse with the nucleus of the egg, and together they will make a joint 46 chromosome nucleus. And this is where fertilization happens. And the minute that one sperm gets in, there is like a barrier that is instantly formed around that newly fertilized egg, and that makes sure that no other sperm are allowed in. And once it's fertilized and it's 46 chromosomes, it is now called a zygote. This is an egg surrounded by a whole bunch of sperm. This is a photo of that sperm that's getting through the coating around the egg. Here's a picture of kind of relative thickness of that coating around the egg, and how the picture of one sperm that's actually been able to fertilize and get into the egg. And once that does happen, there are some stages of that early embryonic development that we're going to talk about. And the first one is the morula stage. There will be M, B, G, and N, the 4 stages of embryonic development. And this one, first one is the morula stage. You will see as I go through these in order that this order is something that will be important to know for like numerical response questions. So before I start talking about them, I can just say that some time ago, some students we had kind of brainstormed about an acronym to remember this by, and we came up with My Baby Good Night. And that is something that over the semesters, I asked people to make some other acronym if they'd like, but we keep coming back to that one. So My Baby Good Night will be the way that you can remember those stages in order. Morula is the first one, blastula, gastrula is the third one, and neurula stage is the last one. So in the next few slides, we'll be talking about what each of those stages is important for, because they're very significant stages of embryonic development. So this first one, the morula stage, is when that zygote starts dividing, and just like you would use a cleaver to chop through a ball of dough, every time this cell is split, it cleaves. It's in the morula stage still the same size as the original egg, and so cell division is happening. The cells are dividing and dividing, but this size of the morula is the same size as the egg was to begin with, and the reason for that is so that it can keep tumbling down through the fallopian tube, because that fertilized egg is headed to the uterus where it will implant and grow for the duration of the pregnancy. So at this point, it's small, it's on the move, and it's at this point when a cell can fall off of it and start dividing on its own and form an identical twin. The statistics here are just talking about how often. In the first 4 days, 20% of the time we get those twins, and in the 4 to 8 days later after fertilization, 75% of the time is when we get the monozygote, one zygote that turns into twins because of a lost cell. And we've got a few other situations where if it's later than that, those twins may be co-joined and still attached somewhere, because at the point of 2 weeks, it's starting to form more body structures. So if a section of it falls away and divides by itself at that point, that's where we'd end up with co-joined twins. On the topic of twins, if it was 2 eggs released that month and 2 different sperm were involved, you can still have twins, but they would be twins that are no different than brothers and sisters, or 2 sisters that were born at different times, so they would be non-identical. Okay, so done with our first stage, the morula stage, and we're moving in to the blastula stage. And the blastula stage is where this blastocyst forms, and it will have this inner cell mass that you see here that is actually going to be the baby. And it's also got this outer area that's called the trophoblast, outer layer of cells, that will be cells that are going to implant into the mom's uterus and end up being part of the placenta, which is an exchange area for mums and babies, nutrients, wastes, and gases. Okay, so what we can see in the blastula stage is that there's some fluid, it's called blastocele, area that's forming and sort of pushing the cells into that inner cell mass onto one side. Here's an actual photo of a blastula. You can see the inner cell mass right there. And you can see the development in this slide of where it was a morula to where now the fluid builds up and pushes the inner cell mass to one side in the blastula stage, and it's also the blastula stage where implantation into the uterus begins. And you can see here the morula stage, but over here blastula stage when you see all the inner cell mass right there. Okay, lots of pictures of the same thing shown different ways. The blastula will start forming kind of like little roots that will implant into the uterus. And so if I look at this photo, it's really a good one to show the uterus, the ovary here, the ovulation of an egg, and how it was fertilized right about there. So it is fairly early on in its journey down the fallopian tube that it could be fertilized. You can see it's about here when it's 2 cells, here when it's 4 cells, here it's about 8 cell stage. Here it's still they're still all the same size. Remember, the morula is the same size of the egg, but it's about when it enters the uterus when it starts changing and becoming a blastula. And right here you can see how it's implanting, and it will implant and start growing, and it's implanting to get that access to mom's nutrients and oxygens and have a place to exchange and get rid of wastes and carbon dioxide into, and mom's blood will take that away. So one thing that's interesting here you can see is that for that first stretch of time, maybe a week before implantation happened, that little morula is not accessing any of mom's bloodstream oxygen, nutrients, or even toxins. So if someone is concerned about getting pregnant while, let's say, drinking alcohol or something, they have to realize that in that first week, it was actually not attached to any source of anything from the bloodstream at all. So about a week later, though, that's when it would be possibly affected by what we call teratogens, which are chemicals that can affect fetal development. And so we'll talk about those a little bit later too. All right, so it's about a week, day 7 here, when the little trophoblast is implanting. And I know this looks like little horns, but that's kind of what the implanted parts of the little blastocyst look like. They call it invasion.